Good morning and welcome back to the course. So we're taking a little bit of a detour here, and the reason for the detour is that we're in the middle of talking about life as defiance. In the context of that, we talked about a variety of different biological batteries, one of which is the transmembrane gradient of ions, for example. If I have a gradient of hydrogen ions, there's many ways to harvest that in order to perform work. And the case that we're using as our, I think, very powerful case study is what are called symporters and antiporters. The idea being that there's simultaneous, like for the moment, think of it this way, as one-to-one stoichiometric transport across a particular membrane protein. And for one of those molecules, it's traveling down its gradient, and the other is traveling up its gradient. And if, in a way, the only thing we care about is the one going up its gradient, such as a sugar being pumped into a cell where there's more sugar than there is on the outside, then effectively what we're seeing, if we play a movie of just the behavior of the sugar, we'll see a movie of Humpty Dumpty going back up onto the wall. It's a wrong way, ostensibly wrong way reaction. And the point is that allied with that is a corresponding down the gradient transport of ions, for example, sodium or hydrogen or so on. So what kind of proteins carry out such activities? And the answer to that question, for my purposes, is going to be, one of the most important concepts in all of biology, and that is, at least at the level of molecular biology, and that is the nature of allostery. I heard it said, I'm not going to say who, but I heard it said, and I probably said this already in a different vignette, that there was a very, very noted biologist, somebody that I admire enormously, really, really impressive person, and a key player in the advent of modern biology. And what he had to say about physicists making their way into biology is that he was unwilling to even engage with them, to talk to them at all, unless they could explain what allostery is. Physicists need not apply. If you can't explain allostery, physicists need not apply. That's heavy-handed. I'm not saying that I necessarily agree with that, but I think it's funny. And I also think it's, as with many sort of caricatures or harsh statements or sort of, what I want to say, cliches, there's a grain of truth to them. And I think that the point being made by this very well-known biologist is that allostery is so ubiquitous and so critical to the functioning of cells that one really needs to think about it. So allosteric molecules, for our purposes, for my purposes here, are molecules that undergo a conformational change, a change of state, let's just say, from an active to an inactive, or from an inactive to an active conformation. And what tilts that balance, so the first thing I want to note is that there is a free energy balance between active and inactive conformations in the absence of any sort of ligand. So the active and inactive conformations, as you see in the top with that little caricature of Nigel Orme and me, our faces plastered onto a repressor molecule, is that the molecule is going back and forth between the active and the inactive conformations, a bit like the famed Janus, which you can look up on Wiki. So now the point is that free energy balance, so if I think about this, for example, the inactive state might be the one that's favored by quite a bit in terms of the intrinsic internal energy, and therefore when I use Boltzmann statistics, I'll say the inactive state is much more probable than the active state. Now the point is that if the KD, the binding energy, is different for the active and inactive states, it may actually be that the binding energy is better in the active state, and that leads to a net swapping of the equilibrium probabilities when in the presence of a ligand. So at the bottom of this page, I give you three distinct examples of this. One of them is a ligand-gated ion channel. So here the driving force for gating is the binding of some ligand. If the ligand's bound, it tends to favor the open state. If it's not bound, it tends to favor the inactive state. There's also enzymes that are essentially turned on and off by the presence of some ligand. So here, in the absence of the red ligand, the enzyme is closed for business and cannot act on the blue substrate. And then there's a number of examples, such as chemotaxis receptors, quorum sensing receptors, and so on, where the receptor is sitting in the cell membrane, and in the presence of a ligand, it is then competent or licensed to effect a phosphorylation reaction, for example. So here I just wanted to quickly indicate to you the idea that this kind of allosteric interaction is absolutely ubiquitous. So at the top, I show you an example of a signaling pathway where there's some sort of exterior ligand that actually turns on a metabolic enzyme. So in other words, there's some stimulus, which we see up here. There's a signaling pathway, the details of which I do not concern myself with for the moment. And the terminal molecule in that pathway is this metabolic enzyme, and in the presence of the effector ligand, it now becomes active. So that's case study number one. Case study number two has to do with transcription factors and the control of transcription. So here you see there's some exterior ligand that leads to a signaling cascade. The repressor molecule, in the absence of the ligand, is repressing. But now, over as you see on the right, in the presence of the ligand, the repressor is induced, it is inactivated, and this means that the gene is turned on. And then finally, the last example that I'm showing here has to do with motility, where there's some exterior ligand. In the absence of the ligand, there's some sort of regulatory protein, which I show here as kind of a ball and chain, and this thing is inactive in the absence of the ligand, and then in the presence of the ligand, it undergoes a conformational change, which leads to local polymerization of actin filaments, just for example. All three of these are examples of the ways in which allosteric molecules are participants in signaling cascades. And to remind you again, the notion of allostery refers to the fact that a given molecule can have its free energy balance between active and inactive conformations tilted by the presence of a binding molecule. I don't know how much more I will say about allostery. I'm sure we'll work out the statistical mechanics of it, but let me just say it's a deep subject. There are different competing models. There's the MWC model, Monod-Wyman-Changeux. There's KNF model, which is Koshland, Namath, and Filmer, if I remember correctly. You know, there was a lot of heat surrounding the difference between these different models. For me, when I'm wearing my statistical mechanician hat, I'm sorry to say, but I really literally think of these as being absolutely trivial differences, barely worth our breath to talk about. But people get really worked up about what the underlying molecular mechanisms are of a given type of allosteric molecule. Manfred Eigen actually wrote down an even more general model. I like that one a lot. I think it's cool. You know, at the end of the day, if you like, the final court of appeal is experiments, but for me, I'm actually interested in intuition. And the difference between MWC and KNF is absolutely a matter of indifference to me as regards intuition. So I just wanted to say, you know, realistically, there's a number of different molecules that come up and that are allosteric. I just want to point out here the nicotinic acetylcholine receptor is an example of a ligand-gated ion channel, which can be described within the confines of these kinds of allosteric models that I talked about. This is a G protein coupled receptor, and there's a number of different examples of this that are super amusing that one can think about. I'm not going to say anything more about that right now. This is the LAC repressor molecule, which is induced, as many of you know, by IPTG. And then the last example is hemoglobin, which undergoes a so-called R to T or T to R transition, tense and relaxed, in the presence of oxygen. So in that case, oxygen is the ligand that induces the allosteric change. So this is a brief vignette. The point of it is just to introduce the idea of allostery as a pretext for being able to think about the case of symporters and antiporters that we're going to talk about now.